Chapter 5 is all about cell division and how do we get new cells. You can see here in this image some beautifully stained cells going through cell division. You can see the DNA in blue. You can see the microtubules that are in that brilliant golden color. These are the learning objectives for this chapter. I'm going to break it roughly in half. So for the first part of this chapter, I'm going to go through these 9 learning objectives, and the balance of them I'll talk about in the second video. We'll have another video in between of someone else speaking about something of which they are more expert than I, so a very interesting lecture in there. So our first learning objective is to figure out how are new cells produced. Well, in order to answer that question, we have to figure out where do cells come from. So no, they don't come from Dr. Rudolf Virchow, but Dr. Rudolf Virchow was a Pomeranian. No, he was from Pomerania, which is in present-day Germany. There he is. Anyway, Dr. Virchow, very famous physician, very famous biologist, but he was one of the first people to come up with cell theory, the idea omnis cellula e cellula, meaning all cells come from other cells. So new cells come from cell division. So when cells divide, that's where the new cells come from, and on in an endless pattern of descent. So here is a fertilized frog egg, or a frog zygote, going through the first few rounds of cell division. Just watch that for a minute. You can see it all starts from one cell. In this case, we saw 4 cells, 8 cells, dividing into 16, 32, so on and so forth. But not always do we see that the cells are contained within this small of a unit. Usually we expect there's going to be some growth and development. So by and large, we can divide cell division into 2 different kinds in eukaryotes. Mitosis is the cell division that is occurring most frequently. That's what we just saw in that frog zygote. And the daughter cells are identical to the mother cell in terms of what organelles are present, how many chromosomes are present, and stuff like that. Just a matter of semantics. We talk about mother cells and daughter cells and sister chromatids. There's no inherent gender associated with these terms. It's just a matter of convention. So for anyone gets concerned about that, just point of clarification. Meiosis is the cell division that allows for a new generation of organisms because the daughter cells have half the number of chromosomes and they are genetically distinct from the mother cell that produced them. So when we go from one generation to the next, meiosis is the way that we can have the same number of chromosomes as our parents because they produced cells that only had half the normal number of chromosomes. And you have 2 parents, and so it restores that number of chromosomes. Prokaryotes, bacteria and archaea, don't do either of these types of cell division. And I'm not really going to talk about binary fission, but just so you know, and it probably might be clear to you when I describe how these processes occur, that it's not a thing for prokaryotes. So what they do is basically they just split in 2. They copy their DNA, and then each daughter cell gets one copy of that DNA. And it's a lot more straightforward because prokaryotic cells are a lot simpler. They don't have a lot of the same issues with having the proper membrane-bound organelles and stuff like that in each daughter cell. So, second learning objective, let's talk about sister chromatids. In order to get there, you have to define a few other things. So, the genetic material, the DNA, is, we call all of the DNA in a cell its genome, or some folks say genome with the accent on the other syllable. So the DNA molecules are packaged into structures called chromosomes. Chromo means color, and some means body, so colored bodies. And they were called that because early students with microscopes would stain the cells, and the stain would most adhere to the nuclei. And when they were watching cell division, they would actually see these colored bodies that would look very darkly colored, dark blue or dark purple or dark red. And it was actually the proteins that were associated with the DNA that picked up the stain, and it made them so darkly colored. But now we know that the chromosomes are made up of both protein and DNA, and that DNA is the genetic material. So prokaryotes, bacteria and archaea, usually have a single large loop of DNA, a circular DNA molecule. It's not a perfect circle, but it's all scrunched up, and it is a loop. Whereas eukaryotes, like you and I, typically have multiple linear molecules of DNA. So chromosomes, we got to talk about a little bit in chapter 3, I think it was, and I showed you this picture of how during most of the time in the nucleus, the chromosomes are uncondensed. They're kind of like cotton balls that have been stretched out. But then when they go to divide, they scrunch down into these structures called chromosomes. And the chromosomes are kind of packed densely together, nice and neatly arrayed, so that the daughter cells can separate them out more easily, rather than having to tease out the right chromosomes from this mess of stuff here. And here's a cell that's going to divide, and you can see here in the nucleus all of the chromatin is condensing down into the chromosomes. So chromatin is what we call the DNA before it is condensed, before it's all squunched down into those discrete units, those lines that we saw. Chromosomes are the condensed DNA when it's ready for cell replication. Somatic cells, which are your body cells, are non-reproductive cells, and they have 2 copies of each chromosome from the mother and the father in them, whereas gametes are your reproductive cells, or your sperm and eggs, and they only have 1 copy of each chromosome. So as far as how different organisms arrange all of their genetic information, humans, we have 23 chromosomes in our gametes, or 23 pairs, or 46 chromosomes in all of our body cells. Chimpanzees have 24 chromosomes in their gametes. Excuse me. Fruit flies have 4 chromosomes. But lest you go thinking that simpler organisms have fewer chromosomes and complex organisms have more, it doesn't exactly work that way. The record holder so far for a number of chromosomes is this adder's tongue fern, which has 630 chromosomes. And that is a crazy number of chromosomes. Hopefully you can appreciate that, given these other numbers. 630, that's just kind of a ridiculous number. And there are reasons for it, that it has this crazy number of chromosomes, which I'm not going to get into right now. But this is what adder's tongue fern looks like. So you can see it's not like a particularly sophisticated plant. So the number of chromosomes isn't what's really important. well, it is important, but it doesn't determine how complex or simple an organism is. As long as all the genes are present in those chromosomes to give the instructions on how to make that organism's body, that's what really matters. So sister chromatids, to get back to the original question, are identical copies of a chromosome. So if we were to look at, say, this chromosome here in this image, if I were to blow that up and show it to you here, what you'd see is that you've got each of these things in this sort of X-shaped structure here. This is a sister chromatid, and this is a sister chromatid, and they are identical copies of that particular chromosome. So each of the daughter cells is going to inherit one of those sister chromatids, which is going to be the copy of that chromosome for the daughter cell. Okay, so next learning objective is what are the stages of the cell cycle? Basically, a cell goes through two stages, main stages. The mitotic phase, when it's going to go through mitosis, the actual cell division, and cytokinesis, which is the division of all of the organelles, the ER, the Golgi apparatus, and so on and so forth. And then the rest of the cell cycle is what we call interphase. So most of your cells in your body right now are in interphase. It's where they spend 90% of their time is just hanging out, doing whatever the cells need to do, being skin cells, being liver cells, whatever their job is. But there's parts of interphase where they may copy the chromosomes to prepare for cell division should they receive the signal that it is time to divide. So another way to represent that is in this sort of clock face pie chart here. So interphase is roughly 90% of the cell cycle. And that's most of the time that the cell is around, it's going to be in interphase. So in interphase, we've got sort of three subphases there. We've got the first gap phase called G1. S is for synthesis, where the DNA is actually copied. And then the second gap phase, or the G2 phase, where the cell is preparing for mitosis, but now it's got copies of all of the DNA. So the cell continues to grow during all of those subphases, but it can't actually divide unless you know that there's accurate copies of all of the DNA. And then it will go through a brief mitotic phase, division of the organelles, and then you've got two cells, and the process will continue.